Phage display libraries for antibody V-region production. This is a technique for producing antibody-like molecules. Gene segments encoding the antigen-binding variable or V domains of antibodies are fused to genes encoding the coat protein of a bacteriophage. Bacteriophage containing such gene fusions are used to infect bacteria, and the resulting phage particles have coats that express the antibody-like fusion protein with the antigen-binding domain displayed on the outside of the bacteriophage. A collection of recombinant phage, each displaying a different antigen-binding domain on its surface, is known as a phage display library. In much the same way that antibodies specific for a particular antigen can be isolated from a complex mixture by affinity chromatography, phage expressing antigen-binding domains specific for a particular antigen can be isolated by selecting the phage in the library for binding to that antigen. The phage particles that bind are recovered and used to infect fresh bacteria. Each phage isolated in this way will produce a monoclonal antigen binding particle analogous to a monoclonal antibody. The genes encoding the antigen binding site, which are unique to each phage, can then be recovered from the phage DNA and used to construct genes for a complete antibody molecule by joining them to parts of immunoglobulin genes that encode the invariant parts of an antibody. When these reconstructed antibody genes are introduced into a suitable host cell line, such as a non-antibody producing myeloma cells used for hybridomas, the transfected cells can secrete antibodies with all the desirable characteristics of monoclonal antibodies produced from the hybridomas. And that's phage display libraries for antibody V-region production.